Hello, everybody. I just wanted to talk about a couple of things real fast before we get into our discussion. The first one is the upcoming exam for the end of the year and what that's going to look like. I've had several people starting to email me about that, and we are still working up the details. And it's a little bit different because we have multiple people involved, we have multiple schools involved, and I mean, with WIT being involved with that. And so just trying to make sure that we get everyone in the discussion that needs to be in that discussion and that what they want to have happen and how we're able to deliver it as a school district right now is something that we can manage and get taken care of. So I am continuing to work on that. As soon as I have that information, I will make sure that I deliver it to you guys, whether that's through a post or through a video. But to make sure that you keep checking Canvas, because like I said, as soon as I get that information, I will definitely pass that on to you. The other thing is the start of the virtual contact meetings with the team meetings on Thursday and Friday, depending on if you are an A day or a B day student. The first week that we met, which was last week, I just wanted to be able to have people log on, get used to the system. If there was any problems, troubleshooting that we needed to take care of, that we had time to do that. It's a little bit different. I know a lot of people have been using Zoom and are more comfortable with that, are more familiar with it. And so with that transition over to using Microsoft Teams, like I said, didn't really want to have anything formal in place. Moving forward over the next couple of weeks, that is how I'm going to plan on building in review time for the final exam. So it will be a little bit more structured than what it was last week. So make sure that you are continuing to check in Canvas. For you guys, I actually posted it as a module, and the reason that I did that is because we have a long list of assignments and I was afraid that it would get lost in there. So starting a module section, it pops up still on that same page, but it's separate in its own little section. And so I was hoping it would stand out a little bit more than if I embedded it in with the assignments. So moving forward, continue to look for it in that module section. I am going to have to make a time change this week with my 1B class. I have my 4 kids here, which we are sharing computers to get everybody on for their meetings that they have. And there's a conflict of times with that 1B and getting my child on. So it will still be within our normal class time meeting, but it will not be at the exact same time as it was last week. So please make sure that you guys are looking for those changes as we proceed into this Thursday and Friday. Keep coming with the emails if you guys have questions, problems, issues, and I will get those taken care of as quickly as I can. So from here, we're kind of wrapping things up and moving out of the endocrine system. And if we were still meeting face-to-face, there's a couple of labs, obviously, that I would have loved to have done and just don't have the ability to do that. But the endocrine system, with it being your hormones, there are different activities that we have where we fluctuate those, and you guys can actually see those changes within your own body, and makes it a little bit more realistic. So not being able to do that, we're, like I said, kind of moving out of the endocrine system and making our way into our final chapter, which is the reproductive chapter. So at this point, we have covered 10 out of the 11 body systems and kind of wrapping things up. Now we do have the male and female, and we do a little bit with pregnancy as well. So that will be the discussions that we're covering as we move forward. Now, the packet that you guys are going to have actually starts in a little bit different place than the actual reproductive systems. And I want to go back to a concept that we talked about very early on in the year. And honestly, you guys should have had this multiple times as far as the discussion on cell division. I know you guys get it in middle school, you get it in Bio 1, Bio 2 if you took it. You would have also heard of cell division. And then at the start of our year. But when we move into this particular chapter, we start talking about a different type of cell division. Now, all of your body cells, the somatic cells, are going to go through mitosis. And that is a type of cell division in which you are making an identical copy of the parent cell. So one cell division, the original cell divides and becomes 2 new cells. They are identical to each other. And we use the process of mitosis when we are trying to get more cells because we're growing and developing. We are trying to replace cells that have died, or the cells are just getting too big and we need to reduce their size so they can continue to function in a proper manner. So the goal of mitosis is very different, and I just listed those for you. On the other side, we have meiosis. There are only 2 cells in the entire body that use meiosis, and if you break it down by gender, there's only 1 cell in the female that uses meiosis, and there's only 1 cell in the male body that uses meiosis, and those are our gametes. So the egg cell in the female and the sperm cell in the male body. Now, the reason that we have a different process for the gametes is because the goal is different. We're not trying to replace cells that have died. We're not trying to produce cells because we need to grow and develop. We are trying to take the cells that we need for reproduction and prepare them so that they are ready if fertilization would happen to produce offspring. So, a couple of things do need to happen in a little bit different manner because of that reason. Now, if you look at that first slide, it kind of talks about some of the things that I've already covered. It also identifies the fact that the sperm cell is going to be the smallest cell that we have in the human body, and the egg cells are going to be the largest. Going back to that idea or that concept that you guys have heard me say time and time again, nothing in the human body happens by chance. If something is positioned somewhere, if something is a certain shape, there is an absolute reason for it. So when we put that idea out there that you have the smallest as well as the largest cell, when we talk about the sperm and the egg, there is a reason for it. Now, with the sperm cell being the smallest cells that we have in the body, that is because of the number of sperm cells that have to be produced in order to increase the odds or the likelihood of pregnancy. So when the sperm are introduced to the female body, they have some obstacles working against them. Number one, fertilization actually takes place in the oviducts or the uterine tubes of the female. It does not happen within the uterus. So the sperm actually have to travel from where they are initially placed in the body to where they actually are going to be able to locate the egg and cause fertilization. A lot of the sperm cell won't make that journey. They will get lost along the way. They will not have enough energy. They'll die off. And so to make sure that the likelihood of a pregnancy is there, you have to introduce millions of sperm into the female body. So because of that sheer number, having them be extremely small just allows that to be able to happen. Now, on the female side of things with the egg cell, once the sperm are introduced to the female body, like I said, they have to travel through the uterus. But if you think of the conditions of being inside the female body, it's dark. They have no idea where they are going. And so to be able to locate this egg cell, having it be a very large size, again, increases the likelihood that the sperm will be able to find it and cause fertilization. Now, the egg cell is actually so large. It is the only cell in the human body that if you had just one single cell, you'd be able to see it with the naked eye. Only cell that works that way. So if you move on to the second slide, it gives you a visual of what happens in meiosis. Now, mitosis, one cell division. You start with one cell, it divides once, you have 2 cells at the end, like I said, identical to each other. Meiosis has to be a little bit different. In order to accomplish the goal of meiosis, which is to create genetically different cells and also to reduce the chromosome number. So we're trying to achieve 2 things with meiosis. So in order to get that done, the cell is going to have to go through the cell division process 2 times. So you start with one cell, it goes through the first cell division and will produce 2 cells. Those 2 cells will go through the process a second time, and when they divide, you end up with 4 cells at the end, and you can see that depicted in the image there. Now, a couple of things, as I mentioned, you have to have cells that are genetically different from each other from the beginning of the process to the end of the process. And I'm not going to go through what happens in each phase for prophase, metaphase, anaphase, and telophase. That's pretty much the same in meiosis as it is in mitosis. But the one thing that I do want to identify that happens in meiosis is the process of crossing over. So when your chromosomes line up next to each other, in meiosis, what will actually happen is the section of one chromosome will actually break off and exchange part of itself with the chromosome next to it. And that's how you end up with these genetically different cells from the parent that starts it out to the cells that you have at the end, because those chromosomes will break off pieces of themselves and swap them out so that the chromosomes are different from that parent cell. And I have a visual on that, so just hang tight for a second. Now, the other thing that we have to accomplish, and this is really what happens in that second cell division. So if you look at the slide that talks about meiosis 2, and again, when you go through prophase, metaphase, anaphase, and telophase, pretty much the same exact thing that's going on in mitosis, pretty much the same things that are happening in meiosis 1, or the first cell division. But what happens at the end of that second cell division is we take the number of chromosomes and we now reduce it so that we only have half of a chromosome set. Typical human is going to have 46 chromosomes, or 23 pairs. And what happens is we know that eventually the sperm cell and the egg cell are going to unite. If our sperm cell had 46 chromosomes and the egg cell had 46 chromosomes, when they unite, we get 92, which is way too many. So through that process of meiosis, we take that original chromosome number of 46, go through the 2 cell divisions, and when we get to the end, we only have 23 chromosomes. So we have half the number we need. So now when the sperm cell meets the egg cell, 23 in the sperm cell, 23 in the egg cell, they unite and we get back to our number of 46. Now, if you go to the slide after meiosis 2, you'll see sets of chromosomes. Now, this is the same set. We're just showing that same set 3 different times. So you have picture A, which shows the 2 chromosomes lined up next to each other. And that would happen in meiosis 1. Now, you'll notice that the 2 chromosomes overlap with each other, and they actually will exchange pieces of themselves, so that by the time you get to the end of that exchange, it's called crossing over, you can see that the 2 chromosomes you have at the end are different than the chromosomes you started with in part A. And that is how we get genetically different chromosomes. This is why you are not twins to, if you're a female, you're not twins to all the females in your family, and if you are a male, why you are not a twin to all of the males in your family. Now, the other piece of this, if you really look at it, and the one thing that I like, the benefit of this is we have the color. If you look at it, 3/4, a little bit more than 3/4 of the chromosome is still the same as set A. There's a lot of information that didn't get changed out. So you always hear people that say, well, I look a lot like my mom, or I look a lot like my brother. Well, there's a very real possibility in that, because a lot of the chromosome information didn't change, but just enough of it did to have there be different traits, features, characteristics between the people in your family so that you are not identical to them. So the next one that you guys have right after that is just another visual showing you how we go from the 46. Now, obviously, they didn't put 46 chromosomes in there. They put 2. But you have those that 1 pair of chromosomes at the beginning. And when we go through the first cell division of meiosis, you can see that 1 chromosome goes to one of the new cells, the other chromosome goes to the other cell. And then we take those through the second cell division, and that is how we reduce the chromosome number down to half. So that's a visual of how meiosis reduces the chromosome number. The other picture that you had was a visual on how the process of crossing over takes place. Now, the next slide that you have shows you, it's almost identical to the first one, but you see 4 little sperm cells at the end of the picture. And this is important because in the process of meiosis, when you get to the end, all the sperm cells that are produced in meiosis are going to be viable sperm, meaning that they could locate an egg and cause a pregnancy. In the female, it doesn't happen that way. you will have 4 cells at the end, but only one of those 4 egg cells that is produced in meiosis in the female could actually result in a pregnancy. So when the initial cell or the egg cell goes through the first cell division, it's not an even divide. One of those egg cells is going to get overloaded and take most of the cytoplasm, take the best of all the materials, and you get this really nice egg cell. The other cell is lacking, doesn't have as much material, doesn't have as much cytoplasm, it's not as big. Those 2 cells then go through the second cell division, and the same thing happens again. That one dominant egg cell is going to take all of the best material. And so when you get to the end of meiosis for the egg cells, you have one kind of designer egg cell, and you have the other 3 that don't have enough material to actually cause a pregnancy. Now, the 3 that would not result in a pregnancy, those are called polar bodies, and they will simply degrade and get reabsorbed into the body tissue. And like I said, don't ever have a chance to result in a pregnancy. That one egg that overloaded with all the good material, that is the one that would actually break free from the ovary and be in a position to actually become fertilized and result in a baby. So twins actually happen because the egg that's fertilized, it breaks into egg cells or balls of cells. And what eventually happens is, for some reason, when that ball of cells breaks apart, it doesn't reunite and come back together. So for that reason, that ball of cells will break apart, should come back together, but when it doesn't, now you have 2 individual balls of cells that are growing, dividing, increasing in number, and that would be your identical twins. In the case of fraternal twins, 2 eggs were able to leave the ovary for whatever reason, and because of that, both of those became fertilized, and that is why the twins would not look like each other. So moving into the information on the male anatomy. Checking my time. So you have the first slide that talks about the male reproductive system, and just kind of a side view, cross-cut of the body. You are looking at all the internal structures and features and how things are positioned in relationship to each other. So one of the things that I want you to take a look at very quickly is, if you look at the position of the bladder, you can see the ureters coming down from the kidneys. They would be transferring the urine down into the bladder. You'll notice that they actually dip backwards and come in more to the posterior side of the bladder, not so much the top side. When you're looking at pictures from the front, it makes it look like they come right into the top of the bladder. It's actually more to the back. The other thing that you can see on this is the urethra. Now, in the male anatomy, the urethra is going to transfer both urine out of the body. You can see with the positioning of the bladder, and then obviously how the urine would be able to make its way out. But it's also going to be responsible for transferring semen out of the body. So it has to perform a dual role, and we'll talk about that a little bit more as we move forward. So this is kind of everything shown. If you need to refer back to it as we move forward and talk about some of the other structures and parts, it's a good reference to have. So the next slide talks about the male accessory organs. We have the epididymis, the ductus deferens, or vas deferens. They get a lot of different names. Ductus deferentia, like I said, it's one of those structures in the male anatomy. We have a couple in the female anatomy that, depending on the doctor you have, you will get several different terms for that one body part. So if I say vas deferens, that's what I'm talking about, because that's what I was taught and what we used when I was going through. Seminal vesicles, the prostate gland, and then the bulbourethral gland. So those are a couple of the structures that we're going to talk about as we move forward. Now, if you find the slide that talks about the structure of the testes. Now, the testes are unique to the male body, and they are going to be where sperm cells are actually going to initially start or be produced and then go through the maturation process. And they will be housed in the testes for a period of up to 60 days before they actually degrade. So if the sperm cell are not used within a 60-day time period, they would not be viable to result in a pregnancy. And so the body will actually break them down and remove them from the system, so that if the male body is going to use the sperm, the hope is that you would be giving the best possible sperm to result in the best possible pregnancy. So that 60 days is kind of the timeline for those sperm cells. If they're not used within that time period, like I said, they just degrade and get reabsorbed, much like the egg cell in the female body would. So they're produced and mature within the testes. And while they're in the testes, they'll actually be kept in a dormant state. And the reason for that is if they're not going anywhere, you don't want them to be using energy. And so while they're in the testes, that whip-like action that you think of the tail of the sperm cell having would not be utilized. So they are kept dormant while they're housed within the testes. The next slide that we have shows the epididymis. And with the epididymis, this is going to be a series of tubes that you have within the male body, which is actually going to start the process of carrying sperm out of the body. So they're housed and held within the testes, and then there's actually kind of a migration process that the sperm are going to have to take when they exit the male body. So the epididymis would be the first place that they would have to go through. Now, you'll notice it talks about them being tightly coiled, and the reason for that is the number of sperm cells that are going to be transferred out of the body, again, is going to be in the millions. So by having those tightly coiled tubes, it allows for the number of sperm cells to be moved, but also maintain that in a very small amount of space. The next slide that you guys have talks about, and again, this is where you're going to see the different names come up, but the vas deferens. So from the testes, they would move to the epididymis. From the epididymis, they are now going to make their way into the vas deferens. And the vas deferens are fairly long. They're about 45 centimeters. If you think about the difference between that, 2.5 centimeters equals 1 inch. So these are going to be tubes that are going to continue to move the sperm. Now, part of the reason for the length is because the sperm cell will actually finish off the very last part of their development and maturation as they are making their way out of the male body. So that length of the vas deferens is actually going to be utilized to allow those sperm cells to finish off that process and make sure that they are ready when they are introduced into the female body. So the vas deferens are actually going to carry the sperm, and at this point they are still sperm cell and they are still dormant. And you'll understand that process here as we continue to move forward. But the vas deferens are going to take the sperm and travel them to the next stage in the process, which is going to be where they meet up with the ejaculatory duct as well as the seminal vesicles. Now, if you find the slide in your packet that talks about the seminal vesicles, a couple of things are going to happen here that are very important. And I've mentioned already that the sperm cell are not active at this point, and you don't want them to be using energy if they are just being maintained within the male body. So once they hit the point of being united at the seminal vesicle, you are going to get a secretion of fluid that is more on the basic side. And if you think of our pH scale, you have the acidic side, you have the basic side. The fluid that's going to be added at this point would be considered a base. You're also going to get an additional fluid that is going to have fructose as well as prostaglandins. Now, the reason for the base is that in order to keep those sperm cells dormant, they are kept in an acidic condition, and that prevents them from being active. In order to neutralize that, we've now introduced that base-like fluid. It neutralizes the acidic conditions, and now we're going to start to see those sperm cells become active. The fructose is going to be their energy supply. I mentioned very early on that the sperm are going to be introduced to the female body. They have to travel through the uterus, travel up into the uterine tubes or the oviducts to find the egg. That's a long journey for those sperm cells. cells. And so in order to make sure that they have the energy that they need, there is that sugar base or that fructose that is going to be involved so that they can use that as they move from one location to the next. Prostaglandins are a series of hormones that are going to be used through this process. And the next part of the process, as everything is moving forward, is the material will now move to the prostate gland. Now, it's not quite semen yet, so we've changed it because it's not just the sperm any longer. We now have the addition of those two fluids from the seminal vesicles, but we still have a couple more stages to go before it would actually be considered to be semen. So the next move is that it would pass on to the prostate gland. So find your slide that talks about the prostate gland. Now, I would also suggest taking a quick look at the picture, the cross-cut picture, because if you look at the prostate gland, now it's cut in half, but the prostate gland makes a full 360 around the urethra. So that is a gland that completely wraps around the urethra. And the prostate gland is actually going to be a body part, much like the ears and the nose, that don't stop growing throughout your lifetime. So with that prostate gland, it will grow through the entire male's life. And it's not uncommon later in life for males to have issues with their prostate because as it grows, sometimes the growth will actually move inward and it will start to close off that urethra. Now, with the two things that we're moving through that, we're moving urine through the urethra and we're moving the semen through the urethra. And so if that starts to get closed off because that prostate gland is growing and pushing inward, obviously moving those two fluids out of the body becomes very difficult. So the position and the location of that prostate gland, like I said, if you take a look at that picture, it will kind of help you see why that would become an issue. Now, the prostate gland is also going to add to the process. And in this particular, I don't know if you can hear my kids. In this particular case, we have another alkaline fluid that is going to be added. And that is to make sure that all of the sperm cells are exposed to that alkaline fluid, that they are neutralized and they can become active. So you're talking about millions of these cells that have to be coated with this fluid. And so that is why we see that second dose come in as they continue to move forward. So we've gone from the testes, the epididymis, the vas deferens, seminal vesicle to the prostate gland, and the next one that we're going to move into is the bulbourethral glands. Now, the bulbourethral glands sit just underneath the prostate, and you can see on this picture the location of them. They're incredibly small, and what they actually do is they are going to react to stimulation, and they will actually secrete a fluid. It talks about it being a mucus-like fluid. It's usually clear in nature, and it would be the first thing that you would see produced from the tip of the penis. And what's actually going to do is, number one, it's going to coat the inside of the urethra, along with the fluids from the prostate gland and seminal vesicles. It will neutralize the inside of that urethra, because if it's transferring urine out of the body, which has a tendency to be acidic, those conditions are not good for those cells, that semen. And so what it will do is it will actually coat the inside of the penis, neutralize any remaining drops of urine that might be present, just so conditions are right and those sperm cells do not become damaged if they come in contact with any acidic urine. It will also exit out of the tip and coat the tip of the penis so that when it is introduced to the female body, that process is just made a little bit easier. So it's a little bit of a lubrication as well for that process. The next slide that we have talks about semen itself. And I already mentioned that initially it starts out just as a sperm cell, and it takes the addition of all the materials from the seminal vesicles, from the prostate gland, and the bulbourethral to actually produce what is semen. So it's really not till the very end, when that fluid is going to leave the body, that it would be classified as being semen at that point in time. because of all the addition of the base-like fluids, it would be a little bit more on the alkaline side, so it would test more as a base. Prostaglandins, like I said, are going to be those hormones that are going to be added as well. And then it talks about, you're looking at about 2 to 5 milliliters of actual fluid that would exit the body, and that is going to be around 120 million sperm cell. Now, that would vary obviously from one person to the next. You'll notice that it refers to it as being an average, so it does vary slightly. Now, the next slide talks about the external male organs. And we have the scrotum, which we haven't talked about yet, and that is a sac-like structure that you have on the outside of the male body, which is going to be where the testes are located. Now, the testes actually start up by your kidneys. So when little baby boys are developing, you have the kidneys, which are found in the mid to lower back, and the testes are going to be pretty much on point with where those kidneys are. And right at the end of development, those testes will start to drop down and move into the scrotum. If you have a baby boy, one of the first things they will do when you have a baby boy or very early, they will check to make sure that that process has actually happened, because sometimes it doesn't, and then there's could be some potential issues or problems with that. So it is something that they will take a look at to make sure that everything has moved where it's supposed to. Once the testes have descended into the scrotum, they are going to not be at the same level. They will be a little bit off from each other. So one will be positioned slightly higher than the other, and that is just for ease of movement and comfort. And you also have a septum within the scrotum, which is going to keep the two testes separate from each other. So we've talked about septums in other places. You have one in your nose, you have one in your heart, and there's also one in that scrotum. And that is just to make sure that you have that separation. If something would happen to one of the testes, that it cannot easily make its way over to the second one, so that everything is done within the bodies in order to make sure that the possibility of a pregnancy and offspring and a generation past your own is going to be able to take place. So that is why you would see that septum within the scrotum itself. Now, the reason that we need this is because the sperm can actually not live or don't do well at our body temperature. So 98.6 degrees is actually too warm for them. By having them in this external compartment, now they are going to be a couple degrees cooler than the actual body, and that is more their temperature where they're going to be viable. And so that's the reason why we have that external pouch. So we talked about the scrotum, you've got the testes, and then you have the penis, which is obviously going to be what is introduced to the female body in order to make pregnancy possible. Within the penis structure, there's a couple things that we haven't talked about yet. You have the corpus cavernosum and the corpus spongiosum, and those are labeled just under halfway on the picture that you're looking at on this particular slide. And those are going to be two sections of connective tissue. Sorry, I'm just checking my time. Two sections of connective tissue. And what happens is you also have blood vessels that are going to be embedded in and around that connective tissue. So when those blood vessels become stimulated, they will fill with blood, and as they become filled with blood, that along with that connective tissue is going to be what helps the penis get to an erection. And sorry, I jumped ahead on the scrotum. I already talked about that. So that takes care of this particular section for reproduction. And like I said, if you guys have any questions, any concerns, continue to email me. We'll get things worked out. And just a reminder, this is a Monday, Wednesday, Friday week, so we will have another assignment and more material posted on Friday. I didn't last Thursday, again, just because of the meetings and being something new. I wanted everybody to kind of have a chance to get familiar with that. But this week, even with the meetings, we will still have Monday, Wednesday, Friday as far as assignments go. All right, take care, and I will talk to you later.